Hello everybody, thank you very much for joining us for this evening's webinar. My name's Katie, I'm one of the equine nutritionists here at Dengie, and we also have Dr. Katie Williams here with us as well. She's Dengie's Technical and Product Development Manager. We are really delighted to be joined tonight by Professor Jacqui Matthews, who is just appeared on your screens there now, and she's going to be talking to us about anthelmintic resistance in equine parasites. So before we get started, I'll just go through the plan and some admin for the evening. So as always, we are recording this webinar and it should be available on our Dengie YouTube channel in the next week or so. There are lots of other recordings on there from previous webinars on a range of different topics, so do have a look at those if you've not done so already. Just to say, if you are joining this webinar tonight looking to get some CPD points, you do need to watch the webinar live in order to be eligible for your points. To help the quality of the recordings, though, we have got the audience cameras and microphones switched off. But if you do have any questions, please do pop them in the Q&A box, and we'll be answering these between us sort of behind the scenes. But we should also have some time for some questions at the end of the session as well. At the end of the session, we will also send over a link to some multiple choice quiz questions, which everyone is welcome to complete if you want to test yourself on what you've learned in tonight's session. But for those of you who are AMTRA, SQPs, BHS accredited professionals or Pony Club registered coaches and assessors, you will need to complete these questions in order to be eligible for your points. But we'll explain a bit more about this at the end of the session. So that leads me on to introduce our fantastic guest for this evening. So we're thrilled to be joined by Jacqui this evening. So Jacqui firstly qualified as a veterinary surgeon before she completed her PhD at the University of Glasgow. Over the next 25 years in academia, she conducted numerous research projects examining sheep, cattle and horseworms, whilst also teaching at 3 of the UK's vet schools. Jacqui's research has generated more than 150 published papers, and she's authored numerous articles, given seminars, and for many years she was also the parasitology expert on the UK Veterinary Products Committee. She's also a member of several subgroups of the CANTER initiative, or Controlling Antiparasitic Resistance in Equines Responsibly, and this was set up to develop and promote best practice guidelines for worm control in horses. And one of Jacqui's inventions, which was a diagnostic blood test for equine small redworm, was commercialised by Austin Davis Biologics in 2019. And in 2022, she moved to this company as the Director of Veterinary Science, where she now leads the research and development programme, supporting the use of innovative diagnostics for evidence-based worm control to improve equine health. So we're really, really thrilled to have Jacqui with us this evening. So I'm going to stop sharing my slides now and hand over to you, Jacqui.
Thanks so much, Katie. I appreciate that. It's great. I need to shorten my bio, I think. There we go. Just can everyone see the slides?
Yeah.
Take a little minute to go to slideshow. There you are. Great. Well, thanks for joining us tonight, everyone. I can see there's lots of people joined. SQBs, BHS, and hopefully Pony Club people too, because we've Katie's done a great job in getting this webinar accredited. So tonight I'm just going to take a little stroll down the importance of anthelmintic resistance in horses, and I'm going to start by just giving you an introduction to helminths, or as we know them commonly as worms in horses, just a bit of background on what worms horses are exposed to and the relative importance of the different types of worms. I'll then go on to cover a bit about the wormers that are available for use in horses and how worms acquire resistance to those medicines, particularly how resistance is selected in the practices that select resistance. And then I'll turn it around and talk a bit more about how we can understand about helminth distributions and how we can actually use those distributions to affect treatment strategies, which will hopefully help us reduce the risk of resistance. And then I'll just finish off on a bit more information specifically on types of resistance and the different types of worms that we see in horses, the commonest worms. And then finally, I'll just provide an overview of the approaches to reduce resistance risk. And we'll pick up on that next week, because then we will look at those sustainable control approaches for reducing the risk of resistance in horse populations. So I hope no one's just finished their dinner, because it's a bit of a gruesome slide I put up here. But these pictures just depict the commonest parasites that we see in horses. The most commonest worms that we find are the small red worms, which are the cyathostomins, or small strongyles. And these exist as an infection in virtually all horses that graze. And at some point in their life, horses will have infections with these worms, and many horses will have these infections throughout life, either at a low or a high level. The next picture is of the equine tapeworm, Anabocephala perfoliata. And as you can see, this is the inside of the intestines of a horse, and you can see these sort of I think it looks a bit like pasta pesto, these worms that are a couple of centimeters long. And they're a bit different to the cyathostomins, which I just mentioned, because the cyathostomins have a direct life cycle. So horses are infected from eating larvae on grass, and then the larvae develop to adults in the horse after going through this mucosal phase on the intestinal wall. And then male and female worms of cyathostomins develop, and they mate, and the females produce eggs, which then hatch in dung, and then the larvae, the third stage larvae, develop in the dung, and then the larvae come out onto the paddock and the horse ingests them. The Anabocephala perfoliata tapeworm life cycle is a bit more complicated, because these parasites, they require an intermediate host for their development outside the host. So the eggs that go out in dung are consumed by intermediate host irribated mites, and within those mites the larval stage develops, and then the horse would then consume serendipitously that mite from the paddock containing the larval stage cyst, and then it's released when the mite's ingested and digested. And then these worms develop within the intestine as well, but they are hermaphrodite, so there's not males and females. And the hermaphrodite worm it grows these segments from its neck end, and as the segments mature, the reproductive organs fertilize, and then these segments just become full of egg packets. So they're a bit different life cycle. But again, you can see this is quite a large infection in this slide, and you can see the redness, the hyperemia within the intestine, where these worms have attached with their suckers. They particularly like going to live around the junction between the small and the large intestine, and that's quite a tight junction, so they can cause blockages there. And this delightful photograph on the right-hand side is another roundworm, and it's called Parascaris, and commonly known as the ascarid of worms. And it's really a problem in young horses, particularly foals. And you can see these large noodle-like worms. They live in the small intestine, and you can imagine large burdens of these can cause impactions and colic and disease. Again, it's male and female adult worms. The eggs are released in dung. These resistant eggs go into the environment, and the larvae actually develop in the egg. So when the dung is kind of broken up, the eggs persist on the pasture and can last from one season to the next to infect foals. It's been quite a nasty infection. And when the eggs are picked up, the larvae hatch out, and they actually go through this migratory life cycle through the liver and the lungs of the foal, causing damage to the gut, and then they're coughed up and swallowed and go back to the intestine where they mature to adults. The other types of worms, which I'll not cover much tonight, are the large strongyles and the pinworms, but they can also cause problems in horses when they are present. Now, what about the presence of these worms across populations in the UK and overseas is quite similar, actually. As I mentioned at the beginning of the webinar, the cyathostomins, or small red worms, are very ubiquitous. They're found everywhere in the world, lots of different species of these groups of worms, and they live in the large intestine of horses throughout the world, and are very, very common. If you look at prevalence studies in different regions, prevalence levels are at 95% and above, so they're really, really common parasites that have co-evolved with horses. Tapeworms are a bit different, and that probably depends on the access that horses would have to the infected irribated mites. And depending on the survey, we can see prevalence levels of less than 10% to over 80%. So when you're dealing with you know, the question of whether a horse has tapeworm or not, you can't really predict it unless you have got either previous test results or you do test that horse for the presence of tapeworm. Parascaris, the ascarid worm, is a common worm that we find particularly on large breeding farms, where it it's present in foals. And in some places, it can become a sort of annual issue in foals. Large strongyles, which are related to the small strongyles, but by their name, you can imagine they're larger. They used to be a huge problem in horses. They were really common in horses until microcyclic lactone anthelmintics were developed in the 80s and 90s. And these worms are very sensitive to microcyclic lactones. And we don't really see these commonly, particularly in horses that have received regular treatments of these anthelmintics. So we just focus a little bit on cyathostomes for a minute. So although I said that infections are really common, what we find is that most horses carry these worms without any signs of disease, and they tend to have relatively low burdens, particularly adult horses on good grazing, well-managed grazing. And what happens in some individuals, particularly youngsters from 1 year up to 4 years old, if they're on poor grazing, they can accumulate very high burdens of these encysted stage larvae within their gut wall. And these encysted stage larvae can build up to several 100,000, if not millions, of worms. And these then, in some animals, they reactivate to develop to adults at once, and they emerge from the large intestinal wall, and they cause a lot of damage when they come out of the wall, and that can cause weight loss, colic, diarrhea, and other signs of intestinal disease. And I'm sure you'll all have heard of that disease. It's called larval cyathistominosis, and unfortunately, it can be fatal in up to 50% of cases. So it's really, really important that we manage burdens in horses to stop these levels of parasites accumulating. And the same can be said with tapeworms and ascarids as well. It's really important. And the main way we can manage parasite burdens is not just to treat horses with wormers all the time, because we know that doesn't work and it's selective resistance. What we need to do is to start managing our paddocks a lot better than we have in the past. And this is a situation that you probably don't want. It's very high stocking density, and you can imagine some horses will put out maybe 50 eggs per gram, some might put out 500 to 1,000 eggs per gram. And if these horses are pooing onto this paddock all day, that's going to be kilograms of dung going onto the paddock. And if it's not removed, the eggs will develop quickly through to the larval stages, particularly in the spring and summer, and be released from those dung pats within about 2 to 3 weeks. And then massive burdens of larvae can build up in the paddocks to infect particularly young susceptible horses. And these are the sort of things you can see. In fact, I've cut it off. There's actually a really good image of diarrhea coming out of that horse's back end. Actually, that's just quickly a typical clinical sign of larval cyathostomas. Also weight loss and edema, because the horses lose a lot of protein from their damaged gut, and then they can't retain the protein or the pressure within their circulatory system, and it pools, and you end up with sort of edema. And you can see the propitial edema here in this poor horse. And this is a horse that actually succumbed to infection. You can see the massive conditions loss in this little pony. And actually, when we post-mortem, this horse presented at the vet school in Edinburgh. It had over 3 million worms in its gut wall. So for many years now, we have actually had wormers that are available to both treat and control these infections. For the roundworms or nematodes, such as cyathostomes and ascarids, we have 3 classes of anthelmintic or wormer. The first class, the benzimidazoles, were released for use in horses in the 1960s, followed by the tetrahydropyrimidines, and the main compound we use is pyrantel in the 1970s. And then in the 1980s and 90s, respectively, ivermectin and moxidectin, which were macrocyclic lactones, were registered for use in horses. In terms of our products that we have for tapeworms, indeed, tetrahydropyrimidines, the pyrantel compounds at twice the dose used for roundworms, are effective against tapeworms. And then there's a tapeworm-specific class, the isoquinolone pyrazines, and the compound that we use for horses to treat tapeworms in that class is praziquantel. Now, when all these compounds were first registered by the Veterinary Medicines Directorate in the UK, they had to prove efficacy both in terms of the number of worms and percentage of worms they killed at treatment or post-treatment, including the types of worms that they killed and the stages that they killed. And they also had to show effectiveness in terms of reducing the fecal egg counts in horses after treatment. And the way to measure that is to take an egg count at the time of treatment before the anthelmintics administered, and then 2 weeks, 14 to 17 days after that treatment. So in drug-sensitive populations of strongyles or nematodes, for example, you would expect a mean fecal egg count reduction after 2 weeks or after fenbendazole treatment of over 90%. And that's the same for pyrantel. You would expect a mean fecal egg count reduction in the horses that have been treated and tested of over 90%. And then in horses that have received ivermectin and moxidectin, these products at the time of licensing were shown to have a higher level of efficacy. And therefore, in drug-sensitive populations, you would expect a mean fecal egg count reduction for both compounds of over 95%. Now, it's also a little bit more complicated than splitting the classes into anti-nematode products and anti-cestode products, because what we find, particularly with respect to the small red worms, is that the different compounds that are licensed for use against nematodes have varying efficacy against the different stages of the small red worm. So what we find is that benzimidazoles, fenbendazole, for example, is actually really only when it was first licensed, had high activity, high effectiveness against the luminal stages of the parasites, and that would be like the larval stages that will come out of the gut wall and the adult male and female worms. But what was subsequently discovered after the compound was registered, that if you applied a 5-day course, a 5-day daily course, as you find in Panacure Guard, of fenbendazole, you found that it actually had a high level of effectiveness against the encysted stages. And these encysted larval stages of small red worm include the early L3, the late L3, and developing L4. As you'll see, sadly, that resistance is at very high levels to this wormer in cyathostomin populations, both here and overseas. So we wouldn't actually recommend the use of this compound for that larvicidal purpose unless you had some information available to you based on fecal egg count reductions in that population of horses to prove that it does have a level of expected effectiveness. The tetrahydropyrimidines, pyrantel, embony, do not have a licensed effectiveness against these larval stages within the gut wall, but just against the luminal stages, and that's a similar case for ivermectin. Moxidectin, which was licensed many years after these other compounds in the late 90s, is quite a complex molecule, and what happens when it's given to a horse orally, it's very lipophilic and it goes into the fat reserves within horses, and then it kind of leaches more slowly back out from those fat reserves, and that maintains a sort of high plasma level of the compound for a more prolonged period than we see with the other anthelmintics. And what we found is probably related to that persistence is that moxidectin does, in sensitive populations, have a high kill effect against all the encysted larval stages. It's licensed for this purpose in the United Kingdom, as well as the luminal stages. So that's just a bit of background against the differential activity. And that's why for many years, people like myself would advise that horses, because we didn't have any tests against the larval stages at that time, that horses should receive a larval treatment with moxidectin in the autumn or winter, particularly if they're at higher risk of developing these large encysted larval burdens. So after the compounds were first registered, the advice was, oh, what we'll do, and they did this for cattle and sheep, so it's not just a horse situation where the parasitologists and the drug companies advise people just to treat horses very regularly based on the egg reappearance period of strongyles in dung. And the idea was if you kept treating horses and you killed the worms within the intestine before the eggs were released, you would suppress egg output, and then you would just eradicate the parasites. And that would be the end of worms. And as you all know, 50 years later, that's not the case. And as you'll see in a minute, what we've done actually is just by giving these repeated treatments time and time again, is to select for worm populations that are less sensitive to that drug. And keep in mind that these sort of efficacy levels that they were looking at when the drugs were first licensed were like 90 to 95%. So there was always going to be a residue of some levels of worms left after treatment. And it's likely that these worms were probably the sort of less sensitive part of the population, particularly with repeated treatments, that were not being killed effectively by the wormers. And if you can envisage that, you know, we've counted small red worm burdens of several million parasites in individual horses, actually leaving 5% or 1% even of that population can still be quite a sizable burden. And then if we just look again at the life cycle, which I mentioned a minute ago, if you do suppress those eggs and dungs, you stop the larval stages developing on pasture if they're nematodes, or the eggs being eaten by arabiatid mites and then developing as larval stages in the mites, and then you break the life cycle. Of course, a much more useful thing you can do now, which I'll come back to next week, is actually to remove the dung before the eggs develop to these stages, and therefore you can break the life cycle that way without the over-reliance of these repeated treatments with dewormers. So we just go on to resistance selection for a little minute. We just cover some of the reasons why worms have been so good at developing resistance to these compounds. First of all, we know that worm populations are very large, and we also know that the genes within them are variable. And that means when you apply a selection pressure, such as treatment, you can actually start selecting worms within these large populations that are genetically less sensitive to the drug, and then these survive the treatment. And with repeated regular all-group treatments over time, what you do is you end up with a population of worms that actually has a higher proportion of drug-resistant worms within it than drug-sensitive. And it's at that stage that we start to see a reduction in efficacy as measured by a reduced fecal egg count reduction after treatment, or sadly in some cases, clinical disease in horses that have received prior treatments. The type of practices that are known to select for resistance include, as you can imagine, as I just said, regular treatments, keep applying that selection pressure on all horses all the time, over time, and select that subpopulation of worms that are less sensitive to the compound. Something that we used to be taught at vet school was you would dose the animals and then you would move them on to clean grazing, and that was making the assumption that you would kill all the worms in the animals and there'd be no eggs. But obviously, clearly, what happens in that case is actually when you dose them and then you move to clean grazing, you're actually seeding that paddock with worms that are actually less sensitive to the drug, the worms that have survived treatment. So we definitely don't recommend to do that anymore. Things like underdosing, giving the wrong anthelmintic compound to the wrong target species and giving it by the wrong administration route, for example, these are all factors that lead to sort of suboptimal levels of the compound being in the animal system, and that will enable these less sensitive parasites that are present to survive at higher numbers. And therefore, these are strongly selective for developing or increasing the risk of resistance in worm populations, because you're having these subtherapeutic levels of anthelmintic compound at the sort of interface of the worms. Now, resistance is a real issue because it's not a sort of phenotypically obvious thing. We can't look at this horse and say, well, here's Jimmy and he's got 50,000 worms and 40,000 of them are resistant. And what we actually find is that the work done in sheep has indicated that by the time you actually detect a fecal egg count reduction that's below acceptable levels, genetic resistance is actually at a relatively late stage. So if you do get a reduced reduction in fecal egg counts post-treatment, that is very concerning. And we can't even look at the parasites at an earlier stage to look at molecular markers that might indicate if resistance is present and at what level the alleles are present within that population, because we actually know very little about the molecular mechanisms of how these drugs work. So you can't really look at the target. For example, with macrocyclic lactones, you can't look at the single target and see if the gene that encodes the target of that molecule has changed and then changed the target molecule. So it's very complex, particularly with macrocyclic lactones. And the only one exception is with benzimidazole resistance, where the clear target is a beta-tubulin gene, and there have been certain single nucleotide polymorphisms that have been picked up in that gene that indicate the presence of a resistance, but there's not been any molecular tests developed for that because it's probably too late, to be honest, for benzimidazole resistance in small red worm. What we found in horses and sheep, that once a population of worms, whether it's small red worms or ascarids or tapeworms, become resistant to a product, then it's likely that resistance will stay there and they won't revert to set sensitivity, and that's certainly been proven for nematodes. Then we have this possible resistance or issue of side resistance, where you have two compounds that are quite similar, like moxidectin and ivermectin, or in ruminants, doramectin, for example, And what the concern is there is that resistance against one of these compounds also induces a level of reduced sensitivity or resistance to related compounds because their mode of action is similar in the worm. And what we also find is worms are really, I mean, they're really quite clever. And what we find in resistant populations is they're actually even just good at increasing the amount of molecules that pump drugs back out of worms. And they just upregulate the genes that express these pumps to just get rid of the drug quicker. So there's all these different mechanisms of resistance going on within individual worms that can make it really challenging to overcome. And the sad news is that there's no new compounds coming to market in the foreseeable future. So that's all quite depressing. We've talked about resistance selection. We've talked about worms can build up in high numbers in some individual horses and cause clinical disease. There is some good news, and the good news is particularly in adult horses that the worms are not evenly distributed within that population. And what we find that within a group of horses, most have a low to moderate burden and only few have many worms. And the reason that is good is because then you can start using diagnostic tools to come in and identify those worms, those horses that have many worms, and then to target those horses with treatments, and then leaving the horses with the lower moderate burdens untreated. So that you're not putting a selection pressure across all the worms and all the horses at the same time. And this distribution, where most animals are have low to moderate burdens and a few have high burdens, is called the negative binomial distribution. And we see that in every single helminth species that's been studied, whether it's in horses or people or pigs or cows or sheep. It's a very typical distribution of infection. So that generally 20% of a group harbour 80% of the burden. So you can just imagine this field here, the horses in the grey are the ones with most of the burden, and the ones in the black have either negligible or low burdens. And what we need then are tools to identify who these grey horses are that have got the big burdens, and then to treat them appropriately. And we'll come back to that next week. And this is some data that we generated many years ago now, and this is where we looked at many samples, fecal samples from stud farms in the United Kingdom, where the horses actually at that stage back about 13 years ago, the horses had been treated very regularly with anthelmintics. And actually what we found at that stage is that very many of these horses, indeed nearly 70%, had egg counts of less than 50 eggs per gram. And just a few individuals within that population had higher egg counts. And what we discovered was that actually most of these horses that had the high egg counts within this population were indeed the youngsters, and that's indicated in this slide here. So you can see with age, the mean egg counts of each age group are much higher in animals under 4 than the rest of the groups overall, although we've got this little increase here because sometimes we do see a reduced immunity to these worms in older horses, particularly if they have sort of metabolic syndrome type diseases. So it's important to monitor all horses for infections and pay attention to their egg output. So with these horses with these greater egg counts, although the egg counts themselves don't actually correlate very well with the level of burden, we do know that these horses are much more liable to have higher burdens, and these are the animals that we would suggest are at greater risk of high worm burdens and at greater risk of clinical disease. So when we're designing control programs around fecal egg counts, it's important to do fecal egg counts in all horses, but where we've got large groups of younger horses, we might have to do those egg counts more regularly and more frequently because the egg counts do come up quicker after treatment and they're generally higher overall. We want to identify these higher egg counts as quickly as possible so we can treat appropriately to reduce the shedding of eggs into the environment. So you can see that actually it's been quite challenging to get the balance right in control programs between avoiding clinical signs in those horses that are more susceptible to those larger burdens, but at the same time not over-treating groups of horses in that you select for resistance, the wormers stop working, and then you end up in a situation where for the horses that truly need those wormers, the wormers don't work. And that's where we do not want to be in that situation for all classes of anthelmintics. And now is the time to act because we have some level of efficacy to some of the compounds in the different worm species that horses are infected with. And I'll come back to which species are resistant to which types of wormers in a minute, and how we can think about how we integrate that into the types of control programs that we institute. So this is just a little cartoon again that explains the use of fecal egg counts in targeting horses to reduce selection for resistance. So as you can imagine, you've got a group of horses here. Most of them have a low to moderate worm burden, and that in many cases is reflected in the sort of egg shedding level that they generate. So you would expect horses maybe in these categories to have 0 egg per gram, or 50, 100, or 150. And then you've got 1 or 2, usually about 20% of the population, maybe a higher percentage if it's a whole group of young animals, that have these higher burdens. These animals are just more susceptible to infection because really their immune response and perhaps their behavioural activities of where they graze. It's a complex mix of genetics, immunity and behaviour. But whatever the reason for them having a high worm burden, these horses will contribute the most to contamination on paddocks. And what you want to do is to treat these horses for 2 reasons. You want to treat them to reduce their worm burden, to reduce the risk of disease, but you also want to treat them based on egg counts to reduce them contaminating the environment. However, if you leave these horses free of treatments that are not at risk of disease and who are producing much lower quantities of parasites onto the paddock, this means that if you treat this horse here in orange, and the worms that survive treatment release eggs, and those eggs go into the environment and hatch and produce larvae, it means that any larvae that are derived from eggs released from these low burden horses will mix with those larvae from the resistant population, and hopefully that will have a dilutionary effect in the genes that are present within the resistant population at the next cycle, because a mixture of worms will be eaten by the horses on that paddock, and then it's hoped that there is more resistant worms will meet with the more susceptible worms, and then the resistant alleles will be diluted. And that's why we propose not just to target horses for shedding to reduce contamination, but also to have this effect of refugia in diluting the resistant alleles within the population that are derived from the treated horses. So just to summarise, non-treated horses worms are not exposed to the anthelmintic, that's these ones, and then worms excreted by the treated horses go onto the paddocks, and then they're diluted by the progeny of the worms from the horses that are not treated. Of course, what we can also do, which we'll come back to next week, is to lift the dung, so we're reducing contamination overall. It will reduce the burdens on all the horses, and that will also reduce the amount of treatments that will be directed by the results of diagnostic tests, but we'll come back to that next Wednesday. So this is the rather depressing sort of global summary of anthelmintic resistance in small red worms in the world. This was a review that was published a couple of years ago by Martin Nielsen, and what he did is he did a retrospective study of 71 anthelmintic resistance projects that were published since the 2000s. And he looked at the geographic area of these, so he spanned 31 countries and 6 continents, and then he looked at the efficacy within these studies of the 3 compounds that are licensed, the 3 classes that are licensed for use against small red worms. And what he found is that in all cases, in the 58 studies that are published, benzimidazole resistance was recorded. In the 37 studies where they looked at the effectiveness of parenteral salts, 92% of those studies recorded resistance. And in the studies, the 57 studies that looked at macrocyclic lactone effectiveness, ivermectin and moxidectin, there was evidence of resistance in 23% of those studies. So it's quite a depressing picture with regard to small red worms. One thing I would say is that parenteral salts were licensed for daily in feed use in the United States, so there's a lot more resistance measured so far in the United States compared to the UK. And we see about 50-50 in terms of levels of effectiveness in populations. So it's definitely a compound worth considering for treating horses for small red worm, but it's important to do fecal egg count reduction tests to assess if the compound is working. Certainly in microcyclic lactone resistance reports up to when this review was published, most reports were of resistance to ivermectin and moxidectin from Brazil, but there were 2 more recent studies from the United States which showed resistance to both ivermectin and moxidectins in horses imported from Ireland. So we are actually seeing now what I would call true resistance as measured by fecal egg count reduction in horses against for ivermectin and moxidectin. So just a little bit more detail about resistance to benzimidazoles. These compounds, as I mentioned earlier, have been available since the 1960s, and indeed the first report of resistance to benzimidazoles in small redworm was as early as 1965. So the compound had not really been registered for that long before people started to pick up a lack of effectiveness, and I think that's because the resistance mechanism is somewhat simple with just these mutational changes in the beta-tubulin gene. And now, as I mentioned earlier, cyathostomes are resistant to these compounds worldwide, with also a reduced effectiveness of the 5-day larvicidal regime also demonstrated. Again, parental salts, they were available since the 1970s, and the first report of resistance to parental was made in 1996, probably because not a lot of people were really looking at it, as probably it had been resistance had probably been present in some populations before that time, and now it's increasingly being reported, and in some regions, as I mentioned, in the United States, as widespread as benzimidazole resistance. And until recently, there were few reports of ivermectin or moxidectin resistance in small redworms, and these were largely limited to single efficacy estimates in small groups of horses, particularly in stud farms. However, a study of 58 thoroughbred yearlings imported from Ireland to Kentucky documented small redworm resistance to both moxidectin and ivermectin in a series of treatments over 8 months. So that was pretty strong bona fide evidence of resistance in those populations, and the concern of not instituting proper quarantine measures because the resistant worms were actually being imported not just from another farm but from another country. However, despite the relatively few reports of full-blown resistance as measured by fecal egg count reduction to ivermectin and moxidectin, it's likely that there is a level of resistance to macrocyclic lactones in small redworm populations on very many pregnancies, and this is often measured as a reduction in the egg reappearance period of the worm's eggs after treatment. This just summarises one study following on from those stud farms where we picked up the low egg counts in the mares that I showed earlier. And after we did the egg count analysis, we offered the farmers efficacy studies. And this is what we found. We found that fenbendazole wasn't working at all. In fact, we stopped using it, obviously for ethical purposes. And then we found this sort of patchy profile with parental, where sometimes, particularly in mares, we get a higher level of efficacy above the cut-off here of 90%, but in other cases we had a lower efficacy. And in most occasions where we tested moxidectin and ivermectin on these farms, we found effectiveness of the wormer in reducing egg counts above the cut-off threshold of 95%, but we did pick up in one case where we were below this threshold, as has been identified in other studies in other regions of the world. And this is a similar study that was performed in leisure horse yards in England, where we looked at fecal egg count reduction tests for the 4 compounds. And again, this ubiquity in resistance to fenbendazole. Parental performed a bit better in this study, with just a few yards coming below the threshold of egg count reduction of 90%. And then we saw effectiveness of ivermectin in all the populations and moxidectin in terms of reducing egg count at 2 weeks post-treatment, but there was one of the yards was a bit on the threshold there. So again, looking at these results, it's important to preserve these compounds as much as possible so we don't end up with this sort of profile with continued overuse. But it's not quite as clear-cut as that, because if you remember, both ivermectin and moxidectin are quite persistent compounds, and when ivermectin was originally registered, it was observed to reduce egg counts after treatment for as long as 19 to 13 weeks after ivermectin administration. And moxidectin, being even more persistent, was originally measured to reduce egg counts at 12 to 24 weeks. That's a considerable period of egg count reduction. But sadly, for the last 10 to 15 years, there have been numerous studies have reported a shortened macrocyclic lactone egg reappearance period, which is indicated really as an early sign of emerging resistance. This is just that study from Foals. So although on the stud farms we were shown levels of efficacy above 95% for moxidectin and ivermectin for all but one of the populations we studied when we actually looked at when eggs were detected or when the fecal egg count reduced below 90% after treatment. We were getting these much shorter egg reappearance periods than we would have expected compared to when the products were first licensed. So there were some of these farms that had they had parental resistance, they had fenbendazole resistance, and they also had a reduced egg reappearance period to macrocyclic lactones, emphasizing the threat of multi-drug resistance at yard level. Not only are small red worms good at developing resistance to anthelmintics, but parascaris species are also very good at doing this. And it's a bit of a reverse profile that we see compared to small red worms, with macrocyclic lactone resistance being much more common in parascaris than to the other compounds. And it was reported as early as 2002 with resistance to ivermectin in Holland, and since then resistance has been reported on many breeding farms in various regions. Benzimidazole resistance in parascaris is less commonly reported, but it is emerging as an issue and is being identified on stud farms, and even in some cases where they've evaluated combination products, for example in New Zealand, with ivermectin, fenbendazole and praziquantel, and we still get reduced efficacy in one farm. So this is a good reason not to use combination products, because we already have a certain level of resistance there to many of the compounds that are available. And sadly, parental resistant parascaris has also been reported in a few studies as well. So again, it's a bit on the tip with parascaris. We've got common resistance to macrocyclic lactones, and we've got emerging resistance to parental type compounds and also to the benzimidazole. So again, it's really important in foals that we take much more evidence-based approaches to parascaris control, and it's also really important that we monitor the efficacy of the compounds that we're giving. And just to get into the scene as well, Oxyurus equi, which I've not mentioned much so far, but this is the pinworm, which I'm sure you've all heard of. It's a nematode that lives at the rectum of the horse, and the females come out and they lay eggs on the horse's perineal area and skin, and it makes it itchy, and then the eggs are placed into the environment to be consumed by another horse. And what has been reported, there's been many anecdotal reports of a lack of effectiveness of compounds against Oxyurus equi, the pinworm. But in terms of looking at effectiveness, in the review by Martin, it was noted that all the studies that had looked at efficacy of macrocyclic lactones against pinworm resistance was reported, but in these cases, sensitivity was still retained against benzimidazoles and pyrimidine. And probably one of the most important findings of the last couple of years in equine parasitology has been the emergence of resistance issues in the tapeworm, Anaplacephala perfoliata. There have been several anecdotal reports both in the EU and in the UK about cases of tapeworm-associated disease in horses that had received prior treatments, and in fact this is one of these such cases. But there hadn't been a measured study, and that's because it's quite difficult to perform efficacy tests for tapeworm, because egg counts are not very representative of the level of tapeworm burden that a horse has. So it's quite difficult to do a test based on egg counts to measure the effectiveness of an anthelmintic compound against that parasite within the host. However, in this recent study from a thoroughbred farm in Kentucky, the authors looked at 56 yearlings that had been given ivermectin and praziquantel or parental pamoate at the double dose to treat for tapeworm, and they looked at egg counts in these yearlings at the time of treatment and 14 days later. And they also took the opportunity to look at fecal egg counts in two groups of mares on this farm that were given ivermectin with praziquantel. And what they found, that the fecal egg count reduction in the yearlings, when they looked at the egg counts they were able to detect in the yearlings at treatment and then 2 weeks later, was only 23%. And the reduction in the yearlings that were treated with parental was only 50%. And it was identified that praziquantel eliminated eggs in only 3 out of 17 yearlings, with 5 yearlings going from egg negative to egg positive after treatment. And in the case of parental, it failed to eliminate the presence of eggs from the 14 tapeworm positive yearlings that were treated. And it wasn't just a yearling problem. What they found in 9 of the 84 mares that were given praziquantel that were positive for anaplocephalid eggs at the time of treatment, 7 of these 9 mares also excreted eggs post-treatment. So although there's no validated fecal egg count reduction test for tapeworm, the finding of cestoid eggs so soon after treatment in several horses is a significant finding. And this is something that we need to address going forward, because we do not want to lose the effectiveness of these compounds, particularly it's because it doesn't take that many worms to cause severe disease. Worm burdens of over 20 tapeworms are shown to cause quite a lot of pathology within the gut wall, which could predispose horses to colic, and horses with larger burdens over and above 20 up to several hundred worms can develop severe impaction colic, and that requires surgery and can sometimes be fatal. So just to summarise here our overview of the findings that I just discussed. First of all, in cyathostomins and small red worms, resistance to fenbendazole is ubiquitous, with resistance to parentel common in some regions, with resistance to ivermectin and moxidectin emerging, particularly as measured as a reduced strongyl egg reappearance period post-treatment. In Parascaris species and foals, resistance to ivermectin and moxidectin is common now, with some reports of resistance to parentel and fenbendazole emerging. In terms of the tapeworm, Anaplocephala perfoliata, there have been recent reports from the US, as I just described, of resistance to both praziquantel and parentel when administered as a double dose. And in terms of the other worms, we know that macrocyclic lactone resistance is very common in the pinworm Oxyurus equi. Now, there haven't been any reports as yet of anthelmintic resistance in large strongyls such as Strongylus vulgaris, and it would appear that this worm is very sensitive, particularly to macrocyclic lactones, and the populations of large strongyls have been driven down to very low levels. But we don't know what the future holds for that worm, and it's a worm that we should monitor for, both in terms of it developing resistance or in the cases where we reduce our macrocyclic lactone use because we're taking more evidence-based approaches to control. We must be aware that there might be a re-emergence of this potentially very pathogenic worm in horses. So really, with no new anthelmintics introduced since the 1980s and no clear prospects of any new actives becoming available, we really need to change the way we control worms, and we must not rely on the use of anthelmintics solely for the control of worms within horse populations. And in that way, our approach, which is summarised here, seeks to reduce that anthelmintic overuse by applying a risk-based approach. And what we want to do in that risk-based approach is identify those horses that are higher risk and to minimise pathogenic stages of worms in those higher risk horses. And that would be, for example, treatments for larvicidal, small red worm populations at the correct time of year in autumn and winter, and minimising pathogenic stages of ascarid migrating larval stages in foals in the summer. But apart from those strategic treatments that we apply, all other treatments should be based on the detection of the presence of eggs and faeces or antibody levels that provide information on the level of worm burden that's present within an individual. And I will come back to discuss that next week. In order to also minimise pathogenic stages within particularly susceptible horses, it's really imperative that we apply excellent paddock hygiene, and that includes regular dung removal, grazing at low stocking densities, and consider resting pastures and using grazing with ruminants, which I'll come back and discuss in more detail next Wednesday. So if we avoid those high burdens in individuals by improving our pasture management and reducing egg contamination using a test-led approach to reduce egg shedding from high shedding individuals, and applying those strategic treatments in those most susceptible animals at the right times, we will avoid those high burdens in individuals in a more sustainable manner and hopefully reduce their risk of disease without increasing the risk of resistance developing. And there's this reliance that we need now to turn more from the use of drugs to the more applied and regular use of environmental control of these parasites, particularly at the stages that are in dung before they get to a stage when they can either be picked up by mites if they're tapeworm eggs, or they hatch and they'll develop as larvae that are excreted, that are released from the dung pat, as in the case of nematodes. And over all of this, with the environmental improvements and the risk assessment, we have to think about targeting, and we target for shedding based on fecal egg counts and use those tests to identify those high shedders to reduce contamination, but retain a certain proportion of the herd untreated to reduce selection for resistance. And we're also now able to target for stages within the horse with the use of antibody detection tests, which measure the level of antibodies to specific molecules released by parasites or within parasites. And then that will give us a measure of the worm burden within those horses by measuring antibody. And I'll come back and I'll talk about these tests next week. So finally, it's important, and we'll talk about this on Wednesday, to assess risk, to get a profile for individual horses in terms of the tests to use, the treatments to apply, and the management to institute, and then use that information and risk to either improve our management approaches or change them, and also to understand what tests to use and the frequency with which to use them. And in that way, what we will hopefully do is get to the situation where we reduce the level of contamination on paddocks by driving down the larval burdens and infected mite burdens that are on paddocks so that the infection pressure is low. And in these cases where infection pressure is low, where good pasture management is applied, less horses are likely to be infected and they are more likely to have lower burdens. And in that case, when these horses are tested, a lower proportion of them will test positive in these tests. And therefore, based on the test results, less anthelmintic treatments will be administered. And that will lower selection for resistance because there'll be less applications of wormer over time. On the flip side, if we continue to have poor management, the infection pressure within the paddock will be high. More horses will be likely to be infected and have higher burdens of parasites, and a higher proportion of those horses will test positive, and that means that more anthelmintic treatments will be administered, and that will provide a higher selection for resistance over time. And if there's anything you're going to take away from tonight's webinar, please just try and remember this little infographic, because this really drives sustainable control, because it gets us thinking about management, contamination, testing, and treatment of the appropriate animals. I'd just like to thank everyone for listening tonight and coming to the webinar. I'll have a look at the Q&A session and see if there's any questions that I can answer in the chat. I'd just like to thank everybody that's been involved in the research teams that generated the data that I put on the slides tonight, and the people that funded those projects. And I'd like to thank Dengie for working with us to provide this webinar tonight. And I hope you all have a good evening. Thank you.
Oh, thank you so much, Jacqui. It was absolutely brilliant. I think we will definitely learn a lot from that. We have had a few questions that are coming in just as we're finishing. So we'll just answer a couple of them now, if that's okay with you. So one question is, do you know anything about the development of any new anthelmintics? I know you said there's not anything coming, but with so much resistance, is this an area of that pharmaceutical companies are sort of working on, or are they not really prioritizing it at the moment?
Yeah, it's as far as we're aware, there are no new compounds being developed that will come to market soon. There was a lot of money put into the development of new compounds in the 90s by some of the big companies like Pfizer, which became Zoetis, and they spent a huge resource, and they came actually out, and it was like a screening based approach that they took. They screened all these different compounds against different nematodes, and they came out with very few targets. And in fact, the only new targets really that came out of that have been anthelmintics that have been developed for sheep and have been marketed in the last sort of 10-15 years, but these have not been marketed for horses. I think in one case, one of the compounds is quite toxic, and the other case, I think there's questions over its broad efficacy against the different stages. So it is looking quite depressing in terms of new actives, and I don't think there's any way we could develop a vaccine, for example, because the infections are so complex that I think it would be really challenging. As someone that I'd worked on vaccine development for a single nematode in sheep, to do it for these multi-species infections, I think would be really challenging. So I think we've got to preserve what we've got.
Absolutely, and I suppose if something new did come to market, we run the risk of getting reliant on that again, and 10 years time we end up in exactly the same position. So
absolutely.
yeah, absolutely. And I think sort of talk about that, as you said in your presentation next week as well, and things that we can do. We have had another question, possibly sort of leaning towards next week's topic, but Jenny's asked, it may just be an old wives' tale, but does having sheep graze in the field help to reduce the risk of infestation?
That's not an old wives' tale, and if you come back next week, Jenny, I'll show you the research evidence that has shown that actually using ruminants as a biological vacuum cleaners really can help reduce contamination, particularly in the first part of the grazing season. So what happens is, particularly with small redworm eggs and larvae that go out in one season, they can persist over the winter. They're quite resistant to environmental conditions, particularly the larvae, the third stage larvae, which are the infective stage. And then what we find is that they don't eat these little larvae. They've just got food granules. And as you go into the summer, these food granules are used up, and the larvae tend to die out. So a really good way to control larval levels of small redworm in paddocks is to leave those paddocks rested for the first half of the grazing season. And a really good way to manage that is to bring sheep on. And there's a study from Holland many years ago that showed the effect of grazing horses on paddocks that were pre-grazed by sheep early on in the summer. And these horses had massively reduced worm burdens because they were necropsied in this experimental study. They had massively reduced worm burdens compared to ponies in matched fields, but were just grazing pony fields all the time. So it's not an old wives' tale. It does actually have some evidence basis, but there are some risks about it, but you'll have to come back next week to find out what those risks might be.
Oh, thanks, Jacqui. Give us a good teaser for next week's session.
It's a bit mean.
Ashley's highlighted something that I think is probably kind of situation that maybe quite a few horse owners find themselves in. So the question is, for the case of horses grazing over a large area where poo picking isn't viable, what would you recommend to help with parasite control?
Yeah, this is a really difficult one, and I think it shouldn't be an all or nothing thing. It shouldn't be black and white. So poo picking is not viable. So I mean, there's a perfect storm there. You've got lots of horses grazing a small area, and actually that's all the more reason to try and remove some of the dung, because the infection pressure is going to be quite high. So I think, if possible, there should be some attempt, particularly even in the sort of spring and summer, when the eggs develop quickly through to the larval stages, if they could be picked, if some of the dung could be picked up at least once a week, that just reduces that infection pressure. The other thing is to do as well, then, if it's really such a bad situation for whatever reason, dung can't be lifted, but I can't really see why it still can't be done. People have got horses, they need to consider the sort of different types of management they can apply, and the sort of effort that includes. So the other thing that can be done in that case would be to do more frequent egg counts, and to make sure that you're picking up these high shedders more quickly, so that there's less eggs going into the environment. I mean, that runs the risk of using more product, but then it reduces the chance of the contamination levels building. And until you do all the egg counts in that group of horses, you don't really know what the distribution of egg counts might be like. You might be lucky. It could be a group of 20 adult horses and the egg counts are really high and only 2 of them. So if you really keep on top of those 2 by frequent testing, then that will reduce risk overall to the whole group.
Fantastic, thank you. That's brilliant. So there's a few more questions in the chat box and in the Q&A box, but I think if you're happy to answer some of those, Jacqui,
Sure.
as we go, and I will move on to my half of the presentation. So thanks again,
Okay.
Jacqui, that was great.
That's lovely. No problem. Thanks, Katie.
So, thanks. I'll just share my slides now. There we go. So that should be on everybody's screen now. Brilliant. So kind of sticking with the digestive health theme for the evening, I'm going to talk a bit more in kind of the sort of broader sense about why fibre is so important for the horse's gut. I think many of you will probably be aware that the horse, as a herbivore, has very much evolved to be eating lots of fibre, whether that's coming from their grass, their hay, or their haylage, for example. But we're just going to look tonight into a little bit more detail about actually why that fibre is so important for them to maintain a healthy digestive system, And we'll just touch on some of the consequences that might occur if they're not getting enough. Now, this is a huge, very, very broad topic. So there will be some parts that I sort of don't get to tonight, but we've got lots more information on our website that we can send over as well. So to start off with, I just wanted to cover the difference between actually what is forage compared to fibre. These terms are generally used pretty interchangeably, and I'm definitely guilty of using them interchangeably as well. The main difference really is that forage is the plant material that's actually consumed by the horse that contains that significant proportion of fibre as the nutrients. So things like grass, hay, haylage, alfalfa, straw would all be classed as forage. And fibre is that structural component that holds the plant together. So this includes things like the cellulose and hemicellulose parts of the plant. And this is broken down by the microbial population within the horse's hind gut. So how is fibre provided in the diet? So I mentioned a couple of options on the previous slide. So things like grass or our conserved grasses, so hay, haylage, or even our dried short chopped or pelleted grasses. We've also got alfalfa, which otherwise known as lucerne, which can be made into hay and haylage. It's not commonly done in this country. We tend to see it more as though the high temperature dried, short chopped or pelleted alfalfa. And we've also got our fibrous co-products, so another great source of fibre in the horse's diet. And these come from sort of high fibre materials that come from the production of other feeds or food. They can sometimes be viewed a little bit negatively, but actually they can be a really beneficial component of the horse's ration. So they've got a really good nutritional value. And from an environmental point of view, they can also reduce the carbon footprint of the ration quite significantly as well. So a great example of this is sugar beet pulp, which is a root crop. The sugar is extracted and goes into the human sugar food industry by British Sugar, and the high fibrous pulp which is left is actually a really valuable, easily digestible fibre source for the horse. And despite the name sugar beet, it's actually very, very low in sugar because most of that sugar's already been taken out. So it means we can make the most of these high fibre products which would otherwise wouldn't have been utilised. So a question that we're often asked on the Dengie feed line is how much fibre does the horse actually need? Well, the answer to that is we don't really know how much fibre a horse needs, but we do know how much forage they need. So the current recommendation is to aim for at least 1.5% in dry matter of the horse's current body weight per day, even for good doers or overweight horses. There was a study which was done back in 2010, which restricted forage to just 1% of the horse's body weight in dry matter. And they did find significant weight loss and without any kind of adverse effects on the horse's welfare. But the criteria they used for welfare in the study was actually based on the horse's behaviour, and they didn't actually investigate any digestive health issues such as gastric ulcers, for example. So with more recent work and the kind of current level that most equine nutritionists would now recommend is that 1.5% of body weight on a dry matter basis. So for overweight horses, what we would need to look at in these circumstances is to sort of make sure they're getting enough amount of forage that they need. We can look at reducing the calorie density rather than the amount being fed. So we're ultimately aiming for more chews for less calories. So this can include using things like a later cut forage, which might be more coarse and have a lower calorie or energy value for the horse. We can look at soaking the hay. We can look at incorporating some straw into the ration, and I'll come back to that in a little bit later as well. We do have lots and lots of information on our website and our YouTube channel on forage for the good doer and sort of how to go about that. So we can send those links over after the session for anybody who is interested to find out a bit more there. So we know how much forage the horse needs, but how much will they actually eat if given the opportunity? Now, as you can imagine, this will be hugely variable, but generally the amount that's sort of we normally expect them to eat would be around 2.5% of their body weight on a dry matter basis. This can be influenced by the type of forage that's being provided, the season, so how much grass is actually available or how easy it is for them to access, and also individual differences in appetite as well. Probably won't surprise a lot of you to know that ponies seem to eat a little bit more than horses perhaps do. So they have been recorded as consuming up to 5% in dry matter of their body weight per day. So, you know, sort of more than double of what they need. So I think we've all encountered those greedy ponies at some point. In an ideal world, we would recommend feeding forage to appetite, assuming that it's not going to result in excessive weight gain in the horse. So for horses who are underweight or sort of generally struggle to maintain their weight, or just kind of maintain their weight very well and don't gain weight very, very quickly, it can be really useful to feed them to appetite. There's sort of no reason to restrict it. To help reduce waste, sort of support the environment and your pocket with your money as well, it can be useful to weigh how much you're feeding the horse and then weigh what's left the next day, just to ascertain what their appetite is and how much they're actually likely to eat. This can also be really helpful if you do have a horse who is losing weight and you want to check how much forage is being consumed, because that can be a really good starting point if you're not quite sure why they've lost weight. It might just be as simple as they're not eating enough forage. So it's a really good place to start. So why do we use dry matter? Why do we sort of refer to this when we're talking about our forage quantities? So the main reason is to give consistency. So all forages will have some aspect of water or moisture within them, even hay, which feels obviously very dry to the touch. And this moisture is basically diluting the amount of fibre and the other nutrients which are being supplied in that forage. So if we don't account for that water content, we could end up significantly underfeeding. The reason I mentioned about using it to be consistent as well is that it does also differ between different cuts. So one cut of hay could have a very different sort of moisture content to another one. So a forage analysis is really the only way to know for sure. But in situations where we don't have a forage analysis available, it's not always an option for everybody. These sort of figures on the screen are the ones that we would often use. So this graph just shows we've got the three sort of main forage types, our grass, haylage and our hay. And we can see that hay's got a dry matter content of around 85% with just 15% water. And then at the other end of the scale, we've got grass, which is usually around 80% water. So it's quite a big difference there. What I haven't got on the slide actually is these sort of wrapped hays that we're seeing more of now. So there's definitely more of a trend to sort of produce these sort of quite dry haylages or wrapped hays, and the dry matter content of those would usually sit sort of somewhere between the haylage and the hay levels there. So now moving on to the horse's digestive system. I think a lot of people perhaps don't necessarily think about the horse's mouth or their teeth being the first part of the digestive system. We tend to think, oh, it's just the bit inside that we can't see. But actually the mouth plays a really important role in the digestive system. The motion that the horse chews will really vary depending on the type of feed that's being eaten. So I think it's not very surprising that feeding chopped fibres or forages encourages more chew time compared to cereal-based feeds or sort of more traditional mixes or cubes. And you can see the figures on the screen there. With the amount of chews that the horse requires per kilogram of hay compared to the number of chews per kilogram of oats. So it's sort of 4 times more chews per kilogram of hay. And as you can imagine, it's sort of going to take the horse quite a lot longer to eat 1 kilogram of hay compared to 1 kilogram of oats. And the other important aspect to think about in the mouth is that is the horse's saliva production. So saliva production in horses is different to in humans in that it's not a reflex response. So for us as humans, if we smell something nice cooking or we're looking forward to our lovely dinner coming up later, we generally produce saliva pretty automatically. In the horse, however, food has to be actually present in the mouth, or they have to be physically chewing in order to stimulate that saliva production. It's sort of often quoted that horses produce around 10 to 12 litres of saliva per day, but actually in practice it's sort of likely to be quite variable depending on how dry the feed actually is and how much chewing is required. As you can imagine, if they're spending more of their day eating hay, they're going to be chewing and potentially producing more saliva than if they're spending a lot of their time eating cereals. There have been studies which have shown that the horse's big cheek muscle, the masseter muscle, is linked to saliva production. So the more active that muscle is, the more saliva will be produced. And we know that eating hay, haylage, chopped fibres are going to be associated with more intense masseter muscle activity compared to eating cereals. So this sort of shows us that if we're going to use more fibre feeds instead of cereals, it's going to increase the amount of saliva that's being produced. So why are we worried about how much saliva is being produced? Why does it matter? So saliva mixes with the feed in the mouth, helps to soften the food, and it basically helps to aid swallowing, keep everything going down nicely. Another difference in humans is that equine saliva contains virtually no digestive enzymes. So it's not until further down the digestive tract that these actually appear to start to break down the food. But saliva does contain bicarbonates, and these have a really important role to basically help buffer the acidity further down in the digestive tract. So essentially, if we're encouraging more saliva production, this is potentially going to be beneficial and helping to reduce the risk of problems associated with acidity, such as gastric ulcers. So after the horse has swallowed, the food will pass into the esophagus, which then leads on to the stomach. Apologies if anybody's squeamish for the picture on the screen, but I thought this was a really nice one just to give you an idea of actually sort of what it looks like and how big it is. So the stomach in the horse is commonly referred to as being about the size of a rugby ball. And it really comprises only about 8 to 10% of the horse's total gastrointestinal tract volume. The horse is a non-ruminant herbivore, which means they've got a nice simple stomach similar to a human's rather than sort of multi-chambered stomach like a cow. Because the horse has evolved to trickle feed, the stomach has got quite a rigid structure, so it can't really stretch to accommodate a large meal. So it's really different to animals like lions, for example, who might make one kill and then eat as much as they like or as much as they can, and then they don't eat for a couple of days. That's absolutely not the case for our horses. So the horse is designed to function best on an almost continuous trickle of feed passing through the digestive system, and this is why we typically recommend feeding little and often. If the meals are really too large, the food will basically be pushed through the stomach and the digestive tract much faster. So we're not getting such as efficient digestion, they're not going to be able to get as much benefit out of it. The only really exception to this is our chopped fibres, which are digested in the hindgut, so we can think of them really like hay, so we don't need to worry quite so much about the meal sizes in this instance. Food generally is going to stay in the stomach for about 20 to 30 minutes, but could potentially be longer, and the horse's stomach really will never be completely empty as long as they are trickle feeding. Because of this, acid is kind of continuously being produced regardless of whether that horse is eating. So another reason why it's important to keep that forage passing through, because that acid will be there either way. So I've just included this little diagram. It gives us a bit more indication of sort of what's what in the stomach. So we've got two distinct regions. So we've got the top part of the stomach, which is the squamous region, and the bottom part, which is the glandular region. And for those of you who are familiar with gastric ulcer syndrome or EGUS, might be aware of the two different types of sort of different syndromes associated with that. So we've got our ESGD, or equine squamous gastric disease, which is associated with ulcers in the top part of the stomach. Or we've got our EGGD, or our equine glandular gastric disease, which is more of an inflammatory condition in that lower part of the stomach. And these are now very much classed as different diseases with different causes or risk factors associated with them. For those of you who are interested in learning a little bit more about ulcers, we hosted a really great webinar with Dr. Mike Hewitson from the Royal Veterinary College about a year ago. And this was specifically about glandular gastric disease, and the recording is on our YouTube channel for anyone who is interested. I definitely recommend it. I just wanted to go into a little bit more detail tonight about ESGD, so equine squamous gastric disease, so that top part of the stomach. So this squamous region in the upper part of the stomach doesn't have any inbuilt protection against the acid which is being produced. Instead, it basically relies on this almost continuous trickle of fibre and the saliva entering the stomach. So as I mentioned, the saliva can help to buffer the acidity, helping to protect it. And the continuous trickle of fibre basically can act as a bit of a physical barrier to the acidity, and sort of protect that layer, the lining of the top part of the stomach. So this is really one of the key reasons why low fibre diets can be such a problem for gastric health. As I mentioned, we know that eating sort of forages or fibre-based diets can increase chew time, can increase our saliva production, so it all kind of comes into play when we are talking about reducing the risk of gastric ulcers. And in addition to this, if we're feeding high levels of cereals, which are very high in starch, this can also increase the level of acidity within the stomach, sort of further compounding the problem. Owners with good doers or overweight horses might often be concerned about feeding ad lib hay or haylage. But as I mentioned earlier, we've recently sort of got more evidence to show that straw can actually be a really good alternative option for these horses, in place of up to 50% of the forage ration. There was a study back in 2009 which looked at the incidence of gastric ulcers in a population of horses, and they found that those who were fed straw as the sort of main or only main forage source were more likely to have gastric ulcers. But the key point here is that straw was being used as really the only forage source, which is not something we would typically do in the UK anyway. But because straw can be a really useful low-calorie forage option for good doers, there was a follow-up study which was published in 2021, in a couple of years ago now, investigating the kind of safety of feeding up to 50% of straw to replace 50% of a haylage ration. And this study actually found no ill effects, and they specifically looked for gastric ulcers. So with that in mind, it is something we are confident to recommend now. So if you do have a good do or an overweight horse who're worried about ulcers, straw can be a really, really useful option to replace up to 50% of their forage. So after we leave the stomach, we then move on to the small intestine. So the small intestine is divided into three main sections, which is the duodenum, the jejunum, and the ileum. And in total, it's around 20 to 25 meters in length. So about the length of a swimming pool, if we've got any swimmers amongst you tonight. Food is roughly going to take around 45 to 60 minutes to pass from one end to the other. This will vary depending on a few different factors, such as like the type of food that's being given, also the meal sizes as well, as I mentioned, with larger meals being pushed through a little bit quicker. And the small intestine is a really important area for the breakdown and the absorption of lots of key nutrients, so things like fats and oils, proteins, our vitamins and minerals, and also our soluble carbohydrates, so our sugar and starch. And this breakdown sort of mainly takes place with the use of enzymes. So for example, we've got amylase there. And the bile from the liver is basically secreted into the small intestine, and this really helps to make the environment a bit more alkaline and allow the enzymes to work effectively. So why is fibre important for nutrient absorption in this part of the digestive system? Well, if we look in a little bit more detail about the structure of the small intestine, it's actually lined with millions of hair-like structures, which are called villi. So you can see the black and white image on the screen there, which is obviously a magnified image of these little villi structures. And then the purpley coloured image is actually a cross-section of the small intestine. So what you might see if you're some food passing through the small intestine, you can see all these little hair-like structures poking in from around the edge. And these villi are really important for increasing the surface area of the small intestine, so giving a really nice area for absorption of nutrients. And if we were to roll them out flat, it would give us a sort of area of roughly 3 tennis courts in size. So it's an enormous area for all those nutrients to be absorbed. And coming onto the diet, well, if we've got high starch and low fibre diets, these can create a more acidic environment within the digestive tract. And this can ultimately, unfortunately, have the result of the villi starting to erode or shrink in size. And this means our surface area for nutrient absorption will then be reduced. So it's sort of not ideal if we're wanting to ensure the horse is getting the most benefit from their ration. So additionally, if we do have high starch diets, it tends to mean that fibre intake is also reduced. So really less is more when it comes to starch, even for a poor doer or for an underweight horse. And if we can utilise fibre as an energy or calorie source instead of our cereals, this will potentially firstly help to reduce the chance of the villi eroding in the first place, and also may help to improve the absorption capacity. So definitely sort of start with fibre if you can, if you've got a poor doer, and sort of don't rely on cereals and the starch part of the diet. In case, for those of you who might not have come across this before, we do have a really useful starch calculator on our website. I popped the link in the top there for you. You basically just enter your horse's body weight, the amount of starch in the feed and the amount you're sort of feeding, and it will tell you whether you're feeding too much or an appropriate amount of starch. It is just a guideline though, so generally speaking, if you can keep the starch as low as possible, that's always going to be better. But if you do have any questions on this, you can always give our nutrition team a call, and we'd be very happy to sort of help you with this or give some advice. So after our small intestine, we then come on to the horse's hindgut. So this is comprising of the cecum, the large colon and the small colon. And the hindgut is really the main site of fibre fermentation for the horse. So it's sort of broken down and fermented by billions of microbial population or bugs which live in the hindgut. And as a result of this process, we get a production of short-chain fatty acids. You might have heard them as volatile fatty acids before, SCFAs or VFAs. And these are absorbed through the intestine and can be converted to glucose or fat for energy use or storage in the horse. Now, this is quite a long process, much longer in comparison to some of the other parts of the digestive tract that we've spoken about so far. So very fibrous material can spend up to 60 hours in the large intestine, so it's not a quick process at all. And as a result of this process, we do also get the production of B vitamins and heat also being produced as well. We'll come on to this in a little bit more detail in next week's presentation for those of you who are joining us next week. Now, what's the problem with too much starch for the hindgut? So any kind of large amounts of starch which have escaped digestion in the small intestine basically are overspilled into the hindgut, and this is where they can be rapidly fermented. This can lead to changes in the composition of the microbial population, especially if there's not enough fibre passing through as well. And this can ultimately lead to the death of the bacteria there, which if they can't tolerate those more acidic conditions. And this is known as dysbiosis, where there's basically a disruption to the microbial population. As a result of this, we can get the release of endotoxins, which can cause damage to the lining and sort of open it up potentially to more pathogens escaping from the gut into the body. Into the bloodstream, and this can lead to problems such as laminitis, for example, or otherwise generally poor health. Ongoing dysbiosis can also contribute towards weight loss as well. So if you have a horse who's losing weight, it's worth having a look at how much starch is going into their ration. It might be worth reducing this just to help make sure we're not kind of upsetting the digestive tract anymore. And dysbiosis can also lead to problems such as colic and diarrhea or loose droppings as well. I haven't gone into too much detail about colic tonight. That's a whole other topic in itself, but we do have previous webinar recordings on that subject, as well as lots of information on our website. So how does fibre intake affect the microbial fermentation within the hindgut? So the main short-chain fatty acids produced in this process are acetate, propionate and butyrate. And the type of diet that the horse is consuming can affect the proportion of the different fatty acids which are produced. So there's been a study which has shown that high fibre diets promote greater production of butyrate and acetate compared to a high starch diet. So with a high starch diet, you get high levels of propionate instead. Now, butyrate in particular is a really important energy source for the cells which line the gut wall, which are known as colonocytes. And so it's therefore really important for maintaining that function of the intestinal barrier. And again, keeping it functioning properly and reducing the risk of any pathogens escaping into the rest of the body. And from our hindgut, we then move on finally to the horse's rector. This is about 30 centimetres long, and it's a storage area for waste material before it's excreted by the horse. And what comes out can actually be a really good indicator of what's going on the inside. So looking at things like frequency, how many droppings your horses are producing. Are they a normal consistency or are they a bit loose? Are they a normal colour? Are they a normal smell? And have you noticed any longer pieces of fibre in the droppings as well? So there has been some previous studies which have suggested that pieces of fibre more than 3.6 millimetres in length may indicate dental abnormalities. So if the horse isn't chewing the fibre at the start, they're not going to be basically extracting the benefits as it's passing through the gut, and these long pieces of fibre can come out in the droppings. So if you're doing all your poo picking to help with your sustainable worm control, then it's also a great opportunity to have a look at what you're picking up and make sure it all looks normal and healthy for your horse. So following on from the point about the longer strands of fibre in the droppings, I just wanted to touch on some dental health sort of points to consider. As I mentioned, if we've got poor dentition, this can potentially lead to inefficient digestion and a reduction in that fibre passing through the gut. So these days, horses are generally living much longer, which is fantastic, but we are seeing more and more horses with dental problems, although discomfort in the mouth can actually affect horses of any age. The mouth and the teeth, as we know, are the first part of the digestive system. So if we do have problems here, it can then create further problems down the line. So it's really important to make sure your horse's teeth are checked regularly. As well as those longer strands of fibre in the droppings, other signs that your horse's dental health might be reduced can include quidding, where they sort of ball up and spit out partially chewed bits of forage, or they might simply have a reduced appetite where they don't want to eat as much because it hurts or it's uncomfortable to do so. And these things can actually be really tricky to spot if they're turned out with other horses. It's not something you might notice very easily. So we know that our forage feeding rate should be 1.5% of their body weight on a dry matter basis as a minimum. That's the minimum amount they should be consuming for good gut health. So if they're not able to eat that much, firstly, it can have disadvantages for gut health, but it can also affect their energy or calorie intake as well. There was a study which was done in 2007, which found that horses with poor dentition were consuming about 2/3 less forage in the same time period compared to horses with normal dentition. So if a horse was consuming 2 kilograms rather than 6 kilograms of an average hay, their energy intake or their calorie intake could be around 40 megajoules lower just in that time period. So it's not really surprising at all that horses with poor dentition can be more prone to losing weight as well. So if a horse is losing weight, getting their teeth checked again is a great place to start. The biggest thing to remember here is that horses with poor teeth might need more time to eat, or they might need sort of easier to chew forage. So things like short chopped or pelleted fibres like grass pellets, for example, can be great alternatives. If they can manage pasture and if it's appropriate to do so, so for example, as long as they're not sort of very prone to laminitis, turning them out on grass can be a great option. It's generally, they can sort of manage this for longer compared to sort of conserved hay or haylage. For horses who can manage to chew to some extent, then chopped fibres can be a great option. So alfalfa grass, straw blends can be really a nice option as partial or complete forage replacers. And these can be provided, like in the picture on the screen there, in a nice big tub truck in the field or in the stable for them to nibble on, just like a hay net in a bucket, really. Some horses will unfortunately even struggle with the chopped fibres, so for them they may need to move on to a fully soaked fibre diet. So things like grass pellets, alfalfa pellets, or sugar beet can be really good options as sort of partial or full forage replacers here. And we do have a great webinar on our YouTube channel on sort of feeding and managing the veteran horse. Lots more top tips on this there. So where can you get further information? So as I mentioned, we've got lots of options on our YouTube channel. We've also got recently updated our website as well. We've got loads of great articles on there. And you can also contact our nutrition team for free feeding advice and nutrition support. I'll pop the contact details on the screen there as well. So thank you very much for listening. That brings us to a close of our whistle-stop tour of the digestive system. We're also really pleased to have Jacqui back with us for another session next Wednesday, 2nd of April at 7 o'clock. So if you haven't signed up yet, we will just pop the link in the chat box and hope to see a lot of you there. For those of you who are after your CPD points for this evening, thank you everybody who sent your numbers over or your sort of registration. But the main way that you will get your points is by completing the quiz questions, which I think Katie's going to pop in the chat box in a moment for you. And you've got a week, so until Thursday the 3rd of April to complete these for tonight's session. If you're attending next week's session, we'll send you a fresh lot of questions for next week and you'll have another week to complete those. But for tonight's session, we've got until Thursday the 3rd of April. We will also send you the link on email tomorrow as well if you don't quite catch it from the chat box tonight. For those of you who are AMTRA SQPs, we will log your attendance and points with AMTRA directly as long as you complete those quiz questions. Just make sure you tick the relevant boxes at the bottom of the form or fill in your AMTRA number so that we know which organisation to sort of submit your points to. For BHS accredited professionals, please make sure you complete your normal BHS CPD feedback form. Again, we'll send the link on the chat box via email tomorrow, just in case you don't have access to this or you've not used it before. This will be what you'll need to fill in to get your points after you've completed the quiz questions. And for Pony Club registered coaches and assessors, we will send you your evidence of attendance via email. Make sure when you complete the quiz questions, you tick the Pony Club box, otherwise we won't know to send you this. I hope that all makes sense. As I said, we'll pop it all in an email for you tomorrow as well. But everyone is also welcome to complete the questions, even if you aren't needing CPD points. So I'll stop sharing there. Fab, thank you very much, everybody. And thanks, Jacqui. If anybody's got, I don't know how we're doing with the questions, if we have got any.
Well, I realised I answered them, but I didn't actually, I selected the panelists. I hadn't realised I could go down and select it, so I can answer quickly the questions that
Oh, fab.
Sorry about that. I just
No,
realised
that's okay.
I'm scrolling, I've been answering. Thanks
That's
everyone for the
right.
really positive comments. I'm glad you've all enjoyed tonight. It's great to spend Thursday nights talking about worms.
Yeah.
So here we go. Somebody asked, when ideally should we give insisted dose to horses? There's a lot of confusing information. Well, so the answer is it's important to assess risk, and if the horse is at a high risk of insisted larval burdens, it can be given a larvicidal dose from late autumn through winter. So the whole frost thing is just a myth, really, because actually I get asked this every week. So basically the parasites go into the paddock and they can survive in the frost anyway, and then they'll perpetuate. So the main thing is if there's a horse at high risk of a larval burden, you should treat it at the end of the grazing season to kill those worms, and then assess its risk going onwards. Something that you can do, we've developed a little tool, it was actually on one of my slides, but I didn't have time to talk about it today, was whatsyourwormrisk.com. And that's a free online tool that you can click on. There's 9 questions that either if you're a prescriber or a horse owner, you can work together, answer the questions that are specific to that horse about its own signalmen, its grazing patterns, its test history, its grazing history. And then the answers to those questions will go into a little algorithm, and the result will come out as low, moderate, or high, along with some helpful pointers about what to do next. So if you're looking to give a horse a larvicidal treatment, check out what's your worm risk, and that will give you moderate, low, or high category. So I would recommend at the moment that horses that would come out in the moderate to high category, either like young animals on grazing where dung isn't removed, or they've had repeated egg counts over 200 egg per gram, then what I would say is to treat those animals, larvicidal treatment. Animals that are identified as low risk, an adult horse maybe on extensive grazing where dung is lifted or it's stabled, you can either withhold the treatment, don't give it because it doesn't need a larvicidal treatment because it's low risk, or you can use a small red worm blood test, which I'll talk about next week. You can use that to confirm whether or not that horse is a burden or not, and whether or not it requires treatment. Are there any ways in development to test for encysted redworm? As far as I'm aware, there's no other tests in development, but in future we might consider adapting the blood test to saliva format, but we've got a lot of work to go before we get to the point where it would be available. So watch this space. We're thinking about the options that would be for that, maybe risk-based options, but that's not something that's going to happen in the immediate future. With imminent resistance, next question, in the future and no drugs, is there any natural remedies that actually work? And I'm not aware of any natural remedies that have proven efficacy in horses against horseworms. There's certainly not anything approved. One natural remedy, of course, is to lift the dung to break the worm life cycles, and that's a good natural remedy for exercise. So that's a win-win situation.
Absolutely, I like that one.
Next question. We don't recommend harrowing because oh yeah, someone asked about harrowing. So we don't recommend harrowing because harrowing, basically the idea of harrowing is you spread the dung out on the paddocks, and then the worms are very susceptible to desiccation in sunny, dry conditions. And unfortunately, we just don't get sunny, dry conditions for long enough in the UK for that to work. So all that harrowing works to do really is to spread those parasites around the paddocks away from the dung pat. And, even if you leave your dung down, horses might naturally avoid that. But all you're doing with harrowing is you're spreading the worms around and the mites and the eggs. So don't harrow if possible. In fact, the Canter guidelines have got all the information provided in the background to these questions. I've written chapter 6, which has got all the information about environmental management. So and some stuff as well on testing. And there's other authors written about the parasites and selecting drugs. So they're really, really, really good resource. They're really directed at prescribers, but I think anyone who's quite educated or knows a bit about horseworms, I think they would find the Canter guidelines really useful, and they can be accessed at canterforhorses.org. And then there's a little question. Yep.
Oh, sorry, I was just going to say we can send the link in the email to everybody tomorrow as well. So
cool. Brilliant. I think there's one final question about mixed grazing with ruminants, because someone was worried that it would select resistance in the worms of the ruminants. The main thing to know is that horse worms don't cross into ruminants. So all the worms I talked about today don't cross into ruminants, and vice versa. Ruminants have got their own intestinal and stomach worms that don't cross into horses. And there's two exceptions. One small one is T. axii. That's a little nematode that lives in the stomach that can infect ruminants and horses, but it's not considered of clinical significance in the horse. The one to be concerned about is liver fluke, and that can infect horses, and both cattle and sheep can be a source of liver fluke, which goes into the environment. SQPs will know all this if they do more than equine. And then the stages in the environment are eaten by water snails, and they develop in the snail, and then they're released from the snail as mericidia, and then become these metasicariasis on the land. And that's an important thing to consider if you are thinking about using ruminants for equine grazing paddocks. So I think it's quite good probably to get a vet involved to do a risk assessment for any animals that are coming onto those paddocks, and to consider the liver fluke risk, and if necessary do the right sort of tests in cattle and sheep that can detect liver fluke. And in some cases where actually It's not considered that cross grazing or alternate grazing with ruminants can be done either due to the topography or where the site is, or because there are sites for liver fluke, snail, intermediate hosts. Then it's not something that you can use. And actually, as the person that asked the question suggested, is to reseed pastures, and that's also a really good idea. And I have suggested that where there's been real issues with resistance, particularly with ascarid. So reseeding pastures is another thing to do.
Fantastic, thank
And
you.
I think that's all. Yeah.
That's great. We did also have a couple of questions coming before the session as well, so I'll just ask those quickly if that's okay.
That's fine.
So one of them is, should horses stay off of pasture after worming with a moxidectin-based wormer? And if so, how many days should this be for?
Yeah, I think it's ideally to leave it off 3 days.
3 days.
Yeah,
Brilliant.
3 or 4 days. Yeah.
Fab. And very good. And what advice would you give to owners who are worried about worming their colic-prone horses?
Yeah, and that's always a sort of challenging one. I think, first of all, if there is any smidgen of a clinical issue, if you're an SQP, I would refer the owner to a vet, just in case there is some sort of clinical issue going on. So if you're an SQP and your customers are asking you about their horse being colic prone, I'd be very careful about the advice you give and maybe suggest they speak to their vet before they use a wormer. I don't know if that's what people want to hear, but that's certainly what I would advise. If that said, I think you need to consider why the horse is colicking and whether it's a parasite related problem or not. And certainly some horses will be a bit colicky when they are wormed, particularly if they've got a higher burden, because you can imagine if the compound is killing the worms at a high rate, then particularly if it's a lot of worms, for example, in the gut wall, you might get this response to it, and the inflammation that's associated with that could lead to signs of colic. But honestly, if you're an SQP and you have a customer that's concerned about colicking in their horse, I would probably refer them on to their vet just to cover yourself.
Yeah, thank you. That's great advice. Yes, perfect. Well, I think that's possibly all the questions that have been answered now. Thank you very much for whizzing through those. I'll just check. No, we haven't had any others. So I think we'll draw it to a close on that note. So thank you once again, Jacqui, for joining us. It's been a fantastic evening, and we hope to see a lot of you again next Wednesday, next week at 7 o'clock.
That's great.
Thank you, everybody. Yeah. Thanks, Jacqui.
Thank you. Thanks, everyone, for coming. Take care.
Bye-bye.
Bye.